In this video, we will discuss about application of bacteriophages in control of bacterial leaf blight infection of rice. Rice is the first staple crop of India and feeds more than 60% population of India. India has approximately 45 million hectares of land for rice cultivation, and the production of rice was 120 million ton in financial year 2021. Rice productivity is reduced due to several biotic and abiotic stress factors. Among biotic stress factor, bacterial leaf blight disease caused by Xanthomonas oryzae pathover oryzae is one of the most serious diseases worldwide. Xanthomonas is caused by gram-negative, rod-shaped, non-spore forming bacteria, and their colonies on agar medium are pale, yellow, circular, and smooth with entire margin. Symptoms of bacterial leaf blight disease includes leaf blight, wilting, crease and yellowing and drying of leaves. Organism enters through wounds and hydatodes and moves to xylem vessels, multiplies and results in blight of leaves. Lesions starts as small water-soaked strips from the tips. Later, the lesion appears as milky dewdrop-like lesions and then rapidly enlarges in length and width, forming yellow lesions with wavy margins along the leaf edges. In young lesions, drops of bacterial oozes out early in the morning. Crescent is the symptom which occurs only in systemic infection in young plants and during tillering stages. In these, leaves of infected plants wilt, roll up, turns gradually from white to yellow to green, and then entire plants finally dies, leading to yield loss up to 50%. So for the management of bacterial leaf blight, treatment includes using chemical bleaching powder in irrigation water, and antibiotic treatment includes streptomycin sulfate and tetracycline spray in combination with copper oxychloride. But due to ineffectiveness of chemicals in controlling the disease, effective and eco-friendly biocontrol methods like phage therapy can be used as an alternate approach to control these destructive disease. So what are the bacteriophages? Bacteriophages are the viruses that specifically infects bacteria and replicates inside the bacterial cells, resulting in lysis and killing of the host cell and release of more and more phage particles. A bacteriophage has typical morphology of head, tail, and base plate, and its genome consists of single or double-stranded DNA or RNA, and which is encapsulated inside the protein coat. Tail morphology of phage belongs to three different classes. One, double-layered contractile tail in Myoviridae family, long, flexible, non-contractile tail in Scyphoviridae family, short, stubby tail in Podoviridae family. The size of phage particle varies between 45 to 100 nanometer. Phage therapy is the therapeutic use of bacteriophages to treat pathogenic bacterial infection. So to control the bacterial leaf blight infection of rice, using eco-friendly and effective method, bacteriophage-based project was started in 2018 at National Institute of Biotic Stress Management. In this project, we have isolated few phages against bacterial leaf blight pathogen Xanthomonas oryzae pathover oryzae. The presence of phage were indicated by clear round plaques and clearance of bacterial growth around strict lines on bacterial culture medium. These phages were highly genus and species specific to their host, Xanthomonas oryzae pathover oryzae, and they are not even having lytic activity to other heterologous bacteria. So on transmission electron microscopy, all the phages were found to have head and tail morphology and belonging to different families of the phage. For efficacy trial in liquid culture medium, 2 flasks were inoculated with BLB pathogen. One of these flasks was inoculated with Phage preparation at concentration of 0.1 MOI, and other flask was kept as control. After 24 hours, it was found that there is decrease in visible turbidity in Phage treated flask as compared to control flask, thus resulting in increase in clearance. And on colony counting, it was found to be 99.9% effective in efficacy trial. So for Phage efficacy trial, in protective efficacy in rice pods, rice plants were infected with BLB pathogens, and one group of rice plant was kept as control, untreated control, whereas another group of plants were treated with Phage preparations, and both the groups were observed daily for 14 days. It was found that there is significant reduction of BLB infection in Phage treated plants as compared to untreated control plants, and its leaves, Phage treated plant leaves, have shown very reduced symptom of BLB building in the leaves as compared to leaves of untreated control plants, in which there is significantly higher wilting and necrosis of the leaves. Thus, Phage-based therapy can be used for exploring the control of devastating bacterial diseases of agriculturally important crops. Thank you.